Here in chapter 9.6, we are going to be talking about media used for bacterial growth. So as we have been discussing things throughout the chapter, throughout the chapters, we're talking about culturing organisms. And so we know that we culture organisms by growing them on agar, growing them in a broth, or in some sort of test tube or agar plate, depending on our application and what's easiest and things like that. And so now we're going to take a look at specifics in regards to the different types of agar plates and different types of broth and things like that that can be used for culturing organisms. So typically the type of broth that is used is based on the nutritional requirements. So what is important to understand about whatever it is that you are studying is what it needs. Now, many organisms can be grown on what's called an all-purpose media. All-purpose media. And an all-purpose media is something that supports growth for a variety of organisms. So supports growth for a variety of organisms. And some examples of this are tryptic soy broth, also called TSB, or just basic nutrient broth, sometimes shortened as NB. So tryptic soy broth and nutrient broth are just general, all-around, all-purpose media. So they contain lots of different nutrients, the different macromolecules, but they're not in known quantities, and there's nothing really extra added. So if there's a particular organism that needs something other than just kind of a general nutrition profile, this isn't going to have it. For purposes of organisms that need a specific nutrient profile, which are called fastidious organisms. So let's just put that down first. So a fastidious organism. A fastidious organism is an organism that cannot make certain nutrients. So cannot make certain nutrients. And so we have to get them to those organisms in the broth or in the medium. So they cannot make certain nutrients and require them to be added to the medium. So you can see that an organism that is fastidious needs certain things. So we would have to make sure that the medium, if we want to grow these organisms, contains those things that it cannot make. So an all-purpose medium is not going to work for this purpose. What would work for this purpose is called enriched medium then. Enriched media. So this is specific to those fastidious organisms. So media that contains certain nutrients, growth factors, or vitamins to promote growth of fastidious organisms. So our enriched media then is going to be, there will be different types of enriched media. So if we're trying to grow a particular organism that requires some molecule, then we would purchase, likely from a supply store, this specific type of enriched media, meaning that it is enriched with that specific nutrient that's necessary. So we also have the term called specialized media. Specialized media. And specialized media is used for the identification of bacteria and are supplemented with different things like dyes or pH indicators or antibiotics. So our all-purpose media is, as its name implies, kind of all-purpose general media that'll work for nearly all organisms, with the exception of those that are fastidious. Fastidious organisms then cannot make certain requirements or certain nutrients, so they require them in the media, and that's when we would use enriched media. Now, in both of these cases, we're kind of approaching it in that we know what organism we're using. And since we know the organism, we know we can use an all-purpose media, for example. Or we know the organism, so we know that it's a fastidious organism, and we need to use the enriched media. Now, if we are using specialized media, this would be an example where we're trying to identify the organism. So at this point, we don't know the organism. And so this is media used for identification of bacteria and are supplemented with dyes, pH indicators, or antibiotics. So specialized media is, as we've discussed, is supplemented with something additional. So sometimes organisms can use dyes and some cannot. Some decrease the pH or increase the pH, and then that would be shown by the presence of the change in the pH indicator. Or antibiotics. Some things can grow in the presence of antibiotics and some cannot. So then that would allow us to determine, at least narrow down what organism we might have if it grows on that particular medium. Another type of medium that's important for growing organisms, especially those that we know, are chemically defined medium. Or chemically defined media. So a chemically defined media is where we know the complete chemical composition of the media. So this is the kind where you would purchase it from a seller, and they would tell you exactly the percentage or gram weight, depending on what you're getting, the percentage of this molecule, the percentage of the next molecule, the percentage, so you know exactly what's going to be in it. And most of these media that we've already talked about are not like that. So if we go back up to this all-purpose media, these are not like that. We don't know exactly what's in them because many of the media, like tryptic soy broth or nutrient broth, is a mixture. It's typically a mixture of something like plant products or a mixture of meat products, or it's some sort of ground up something, and that provides kind of a general all-around nutrient broth or tryptic soy broth. So the chemically defined medium is where we actually know the complete chemical composition. So know the complete chemical composition of the medium. And so the example that's given in the book is easy medium. But there are many, many types that you can purchase. And if you looked at a pamphlet or a booklet for a store or online for the store, there are pages and pages and pages of different types of medium. Not all chemically defined, but definitely lots of chemically defined medium in there. The next type of medium we'll talk about is the complex medium, or complex media. For complex medium, this is where we have the unknown complete chemical composition. So we don't know the complete chemical composition. So this is where I was talking about with the other ones, like nutrient broth and tryptic soy agar, tryptic soy broth. So it has an unknown complete chemical composition. And usually contains extracts and digests of yeast, meats, or plants. So the amounts are undetermined and can vary. So you can purchase one container and it's kind of just mixture. They happen to get some particular mixture of ground up meat that has more of one nutrient than another nutrient, and so maybe your organism grows better on that one than it did on the previous batch that you purchased. Typically, it's not so different between the batches that it actually causes some organisms to not grow on them or to not grow as well. Typically, they're pretty uniform, but they're unknown. We don't actually know the complete chemical composition and how much of each particular item is in there. Again, because it's just extracts or digests of different things, of yeasts or meats or plants, and so we don't know the exact chemical composition. So some examples here are the tryptic soy broth that we talked about, the nutrient broth already, and then brain heart infusion. So as you can tell, it's brain and heart all ground up together. So again, we don't know exactly the composition here. The next one is selective media. And for selective media, this is media that inhibits the growth of unwanted organisms and then supports the growth of our wanted organisms. And so it does that by supplying nutrients that these wanted organisms need or can use, while actually causing the death of unwanted organisms. So media that inhibit growth of unwanted organisms and supports growth of wanted organisms by supplying nutrients. So an example of this would be Macconkey Agar. Macconkey Agar. And in the case of Macconkey Agar, what it does is it contains bile salts and it contains crystal violet. And because it contains bile salts and crystal violet, what it does is it actually discourages gram-positive organisms from growing. Gram-positive organisms have a difficult time growing in the presence of bile salts or this crystal violet. But what it does then is it favors gram-negative organisms. Gram-negative organisms. And specifically, Enterobacteriaceae. And Enterobacteriaceae are also known as enteric organisms, and enteric meaning that they are in the intestines. And since they are in the intestines, this is where we're going to find something like bile salts. So they can grow in the presence of bile salts because, our gallbladder is going to be making bile salts, goes from the gallbladder doesn't make it, the liver makes the bile salts, stores the bile salts in the gallbladder, and then when we eat a fatty meal, then the gallbladder is going to contract, it's going to squirt those bile salts into our intestines along with the liver dripping bile salts into the intestines. So we've got a lot of bile salts going into the intestines, and then it's going to be working its way through our intestines. Well, organisms that are living in our intestines then are going to have to be able to live in the presence of bile salts. So that's a lot of gram-negative bacteria, specifically these enteric organisms. And so they will grow on Macconkey agar, whereas gram-positive organisms, which we don't have a lot of in our intestines, will not grow on Macconkey agar. So you can take a Macconkey agar and you can inoculate it with an unknown sample. And if something grows, or whatever does grow, it's going to be likely a gram-negative organism, and even more likely an enteric organism. So that is a type of selective media. And we have several types of selective media that you'll need to be familiar with in the class, and we'll do that in another lecture. We'll go through each of the different types. The next one is enrichment cultures. Enrichment cultures. And enrichment cultures are cultures that maintain nutrients to foster the preferential growth of a fraction of the organisms present in an inoculum. meaning that if an organism can do something, so the example that is in the text is that if you have bacteria that can break down crude oil, so if you can isolate a bacteria that can break down crude oil, you want to then, when you are culturing these organisms, you want to use cultures that only provide carbon from crude oil to maintain their ability to break down or utilize those carbons from crude oil. So this would be an enrichment culture. You've enriched it, and you're maintaining these nutrients. So you're maintaining its ability for, in this example, to utilize carbon from crude oil. So cultures that maintain nutrients to foster the preferential growth of a fraction of the organisms present in an inoculum. So the example that I gave then, of course, was if you isolate bacteria from crude oil, use cultures that only provide carbon from crude oil. Good. So that's a type of enrichment culture. So again, this isn't the same as an enriched culture. If we go back up here, just to be clear, are an enriched media. So our enriched media, remember, is a media that contains specific nutrients or growth factors or vitamins to promote the growth of a type of organism that can't make that nutrient on its own. So down here, when we're talking about enrichment cultures, we're not saying that it can't make nutrients on its own. What we're saying is we're going to preferentially make sure that they continue to do what we want them to do. So in this case, providing carbon from crude oil. It doesn't mean that they can't use carbon from something else, but we want them to continue to use carbon from crude oil. So we make sure that we enrich the culture with that capability. So then we have differential media. And differential media is media that causes a color change. And so this can cause a color change in either the actual bacterial colonies, or this can cause a color change in the media itself. So the media itself has something in it, so something like a pH indicator, for example. And when we inoculate that media with a particular bacteria, if they, say, decrease the pH, then we see a color change, for example. And it can be a color change in the bacteria if that's how that particular bacteria works on that particular media, or it might be a color change in the media itself based on the pH indicator, for example. So media that causes a color change. In bacteria colonies or medium, and it's used to differentiate colonies. So what that means is that since we can differentiate between different organisms in differential media, that means that when we are going to put organisms on this plate, we can see that some of these organisms are not going to cause a change, and some of the organisms are going to cause a change. So then what we can do is we can inoculate this medium, and then we can pick which organisms we want out of what we see. So maybe we're looking for a particular organism that ferments lactose. When it ferments lactose, we can see that it makes a color change. Since it makes the color change, we're going to grab those colonies that made that color change, and we're going to leave the other colonies behind because those don't ferment lactose, so we're not going to take them with us. Or we can determine if we have multiple bacteria, for example. So if we have an unknown culture, we can then plate that. And then what we can do is we can determine, just using the same lactose example, is that if we put it on there and the entire culture doesn't ferment lactose, now we know that's a non-lactose fermenter, and we can go on to the next test. Or if it does, then we know it is a lactose fermenter, and we can move on to the next test. So it's a way to kind of identify, even if we don't know specifically what organism it is, it gives us more information to then figure out what the organism is. So the way it does this is the bacterial enzyme uses substrates. Oops. Substrates in the medium and produces end products. And these end products are those that are going to be colored. Or another example is they will lyse red blood cells. And we see this in our blood agar plates. So there are particular types of media that have blood in them. And when we inoculate those media, we can see whether or not those organisms are going to lyse red blood cells. And we can determine we can see the different characteristics of the certain bacteria when we look at them, and we can see how much they lyse, or if they do at all, lyse those red blood cells. And that's indicated by a clearing underneath. It actually changes the color of the medium. And there are actually 3 different results from that we'll talk about in a future lecture. So a color change or our blood agar. So the example we talked about was our blood agar. And we can see if we have hemolysis, hemo meaning blood, and lysis, so breaking up or killing those blood cells, bursting those blood cells. And then also we mentioned lactose fermenters. So for example, lactose fermenters are going to ferment lactose, and then when they ferment lactose, they will produce lactic acid. And when they produce lactic acid, that's going to actually decrease the pH. It's acidic, so it'll decrease the pH. And then it's going to turn Macconkey Agar hot pink. And again, that's because, remember, in Macconkey Agar, we actually had a pH indicator. So in Macconkey Agar, if we place bacteria on there, we can tell whether it's a lactose fermenter, whether it's not a lactose fermenter, and that's because of the lactic acid that's produced. If it does ferment lactose, that decreases the pH and then changes it hot pink. So it's a very clear distinction between non-lactose fermenter and a lactose fermenter. So these are the terms that you should be familiar with in regards to our media used for bacterial growth. Now, I do want to mention that there are 2 kind of more specific terms, or more general terms, I guess, but 2 terms that you need to be more familiar with, and that is selective media and differential media. It's important that you understand that some media is selective and some media is differential, and some media is both selective and differential. So recall that selective media is a media that inhibits the growth of our unwanted organisms but supports the growth of wanted organisms. So if we inoculate selective media with a combination of organisms, we're going to see the growth of some and not the growth of others. So we can use this, a lot of times this is used to determine between gram-positive and gram-negative. And so you can even see in the example that we gave with the Macconkey Agar, we're going to actually favor gram-negative, so we can take our, say, unknown organism, we can place it on a selective medium, say like Macconkey Agar, and then if it grows, we'll know that that's a gram-negative organism, and then we can do further tests in order to figure out what gram-negative organism that is. Or if it doesn't grow on the Macconkey Agar, then we know it's a gram-positive organism, and then we can take future tests to know what that is. Now, if we then look at differential media, remember differential media causes a color change. So this is saying, okay, we know that these organisms are going to grow on this medium. And when they do, then we see a color change or we don't see a color change. Now, when something is both selective and differential, that means that we can inoculate this and either it's going to grow or it's not, right? And if it does grow, then it may grow differentially. It may be colored or not colored. And the example that was given here again is Macconkey Agar, which is a very helpful type, because if we have an organism that's unknown and we plate it on Macconkey Agar, inoculated Macconkey Agar with it, if it grows, then we know we have a gram-negative organism. And if we have a gram-negative organism and it also has turned hot pink, then we know that it's a lactose fermenting gram-negative organism. So that tells us a whole lot of information about that organism. And then we can move forward from there, figuring out which gram-negative lactose fermenting organism is it. So again, so it can be either selective or differential, or selective and differential. And then, of course, we have all of these other types of media that we talked about, and you now know what a fastidious organism is. And so generally we start with an all-purpose medium because that supports the growth of most things. So we see things starting off in a tryptic soy broth or a nutrient broth, and then in order to identify them, then we would move on to different selective media or differential media.